Hello, my name is Jennifer Rosenbluth, and I'm a medical oncologist and assistant professor at the University of California, San Francisco. I'd like to thank the organizers for the opportunity to give this talk, reviewing practical applications of organoid culturing to breast cancer research, including a discussion of genome editing techniques. I have no relevant financial disclosures. The mammary gland is an organ that is comprised of multiple epithelial cell types, each serving a different function, and these cells also give rise to heterogeneous types of breast cancers with very different properties. Today, we will talk about cell culturing advances that allow us to grow these cells long-term without immortalization, preserving normal cells and their cancerous counterparts. The ability to preserve, grow, and propagate these diverse cell types is readily suggested by the fact that the mammary gland can undergo numerous cycles of growth and quite dramatic remodeling after puberty, during the menstrual cycle, and notably during pregnancy and lactation. These cells should have the capability to do the same things in culture, but this is dependent on our ability to find the right growth conditions. Although improvements can certainly be made, nonetheless, there's been tremendous progress in our ability to model human biology and disease using 3D cultures. If one correlates the complexity of different model systems with the complexity of human organization, then organoids are a reasonable option because they preserve more complexity and model layered tissues that are comprised of diverse cell types organized in a specific pattern. But organoids are still relatively easy to work with in the lab, as demonstrated by numerous studies that have been published using gastrointestinal organoids, the first organoid type to be developed by Hans Clevers in the Netherlands, as shown on the right. Today, I will give an overview of organoids and their ability to preserve diverse cell types, touch on a few considerations for biobanking efforts, and discuss genome editing and genetic screening applications. Organoid cultures are comprised of three main categories of ingredients. These include progenitor cells, isolated from, in our case, normal breast or breast tumors, as well as hydrogel that surrounds the organoids, and defined factors that support the growth of the breast tissue in culture. The general idea is that these factors are supposed to mimic cues that the cells would normally be receiving from the microenvironment and stromal cells. And these factors allow the breast cells to survive, grow, and differentiate in culture to form multiple cell lineages. I do want to point out that in the breast biology field in particular, the term organoid has actually been in use for many decades. Here it's used to describe fragments of the ductal lobular tissue that are retrieved after tissue processing. And when these fragments are placed on a flat tissue culture dish, epithelial and some stromal cells will migrate from the fragment onto the dish. In addition, early studies culturing breast epithelium described differentiation and organization of cell types in floating collagen gels and laminin-rich gels. Normal breast cells could form acinar structures under some conditions, whereas generally breast cancer cells formed disorganized spheres. These techniques were subsequently refined with the development of culture methods that could yield structures resembling terminal ductal lobular units, or TDLUs, the functional units of the mammary gland. HMEX cells are first grown in 2D culture, then placed into a collagen gel. And in collagen gels that are attached to the plastic dish below, branching-only structures develop. However, in floating gels or gels that are made to float, lobular formation also proceeds. This capability was present for early passage HMEX and was lost by around passage 5. A subsequent study used defined components to generate 3D hydrogel scaffolds. Primary breast epithelial cells were seeded into the scaffolds where they self-organized, resulting in the growth of complex ductal lobular structures over 2 weeks. When treated with lactogenic hormones, these cells produce lipid droplets. And these cultures could be passaged at least one time by dissociating the gel and reseeding cells into a new scaffold. More recently, Norman Sachs and Hans Clevers developed a protocol for culturing breast cancer organoids long-term, and we and others have found that similar protocols can be used to culture normal breast epithelium. These cultures are generated by growing breast tissues in a fully defined medium and embedding them in extracellular matrix, and can be used to culture diverse tumor types of different grades and different receptor subtypes. The tumors do display different morphologies with solid, grape-like, or cystic appearances under the microscope. And normal cultures can generate acinar or branching structures, and I'll show some more examples of this shortly. The basic methodology for generating organoid cultures is to digest breast tissue or tumor down to the level of large clumps of cells. These fragments are then embedded within a hydrogel that solidifies on the plate, and a fully defined medium is overlaid. These cultures can be generated from a small amount of tissue, such as a core biopsy, or from part of a larger tissue collection, such as a tumor or even a reduction mammoplasty. The cells will grow and reorganize in the gel to form three-dimensional structures, and these structures can then be digested from the gels and either analyzed or re-embedded for serial passaging. In our experience generating a biobank of normal breast organoid cultures, we found that these organoids formed diverse structure types, including branching structures and structures with central lumen. And we confirmed that these organoids express both basal and luminal cytokeratins and can express the estrogen receptor and grow in response to estradiol. We also found that we could sort these different cell types from culture, separate and then regrow them as organoids, and then analyze them again, basically determining the differentiation potential of each mammary lineage within the organoid culture. We further performed mass cytometry or cytoff using a panel of markers associated with breast development and tumorigenesis. And analysis of these data with examples shown by heat map here, where each row is one cell, and you can see the expression levels of all these different markers in our panel. And these data confirm that organoid cultures were preserving the three major mammary epithelial cell lineages as compared to the tissue from which the organoids were derived. And they also preserved much of the heterogeneity of the parental tissue. This also seemed to be true to a greater extent than other existing culture methods that we were using in the lab at the time. We subsequently performed further graph-based clustering methods using a larger cytoff dataset of breast tissues and organoid cultures, and found that we could determine subsets of each of the mammary lineages, and more formally, identify subsets of cells. So for example, identify subsets of basal cells with higher expression of cytokeratins and signatures of contractility. Basal cells here are divided into these two BA1 and BA2 groupings. And in general, we found that all the cell subsets that we defined could be detected in organoid cultures derived from these tissues, albeit at varying ratios. So this confirmed preservation of diverse cell types in the organoids. Furthermore, I'll just say briefly that we found that we could alter the distribution of cell lineages in culture by removing different components of the organoid medium. So one can use the medium and the fact that we know the factors that are present in the medium to preserve multiple cell types, but we can also adjust the medium to enrich for a cell type of interest, for example. We turn now from normal tissues to tumors. Studies have investigated the ability of organoid cultures to preserve the different breast cancer subtypes as well. And as you can see, not all tumor samples are able to be converted to organoids. This shows the success rate of establishment of organoids from these tumor subtypes. Multiple labs have reported, though, that their efficiency does improve as they gain more experience and practice with whatever type of sample they're receiving. In addition, this graph on the right shows the success rate of marker preservation. And as you can see, if the tumor was ER positive, most organoids are ER positive. If the tumor was negative, ER negative, most organoids are ER negative, and so on. But the concordance is not 100%. And there can be multiple potential reasons for this, including spatial heterogeneity in the tumor with only one region sampled to generate organoids, and possibly changes that occur during the course of culturing in some cases. The slide on the left here depicts examples of marker preservation in organoid cultures as compared to the tissue of origin. And on the right, you can see another report of success rates of tumor generation across the different subtypes. I believe there's been some variation between different labs in this regard. In some cases, groups report highest success rates with ER/PR positive breast cancers. And in other cases, groups report higher success rates with triple negative breast cancers. And perhaps small variations in media components or technique or type of tissue material received, such as a biopsy versus a surgical sample, may underlie these differences. There have been further studies of intra and intertumoral heterogeneity and the extent to which this can be modeled by organoid cultures. though also many remaining questions in breast organoid research in particular. An example of one method for studying heterogeneity was the study on the left in colorectal cancer, where organoids were generated from multiple regions within one tumor. Organoids were then developed from single cells from each region. DNA sequencing revealed mutations that allowed the authors to build a phylogenetic tree for each tumor, and importantly, they could then compare these results with sensitivity of the different clonal organoids to an array of clinically relevant drugs. The drug response could in some cases be correlated with specific mutations in that subclone. On the right is an example of a related concept applied to breast cancer organoids. The authors similarly derived organoids from single breast cancer cells, analyzing 823 individual organoid structures from 52 breast tumors. And each column shows the individual organoids from one tumor. They developed an assay to measure invasive potential, roughly demonstrated by this shape of the organoid in this schematic. And they showed heterogeneity in invasive potential within tumors, and then correlated this functional property with markers such as cytokeratin 14, assessing heterogeneity of invasive phenotypes in cancer cells within as well as between tumors. Based on these studies, as well as many others, I think we can formulate some practical considerations for those who are thinking about undertaking organoid biobanking efforts. First, given heterogeneity, it can be useful to obtain tissue samples from multiple regions of the tissue if possible. Second, preserving tissue for histologic assessment from the region that is directly adjacent to the region you're using to make organoids can be useful, so you can assess properties of the tissue in that vicinity. Long-term culturing potential can vary, so this is something that needs to be optimized and can take some time. And in addition, tissues can be viably frozen for later organoid generation, but efficiency can decrease, and this is also something that needs to be optimized. Heterogeneity can also affect your sample size. And finally, if you're interested in the microenvironment, then there are adaptations that you can make, such as air-liquid interface systems and co-culture systems, such as work by Dr. Muthuswamy that I believe he will present later in this workshop. We're now going to focus on more practical applications for organoid cultures or biobanks, and that is one practical application is genetic engineering. These approaches allow one to introduce genes of interest, reporters for tracking, or mutations of interest into organoids for further characterization. Typically, the components necessary for this are introduced into organoids by viral transduction, or naked DNA transfer can also be used. Furthermore, really increasing reports are utilizing benefits offered by CRISPR-Cas9 to make genetic changes in organoid cultures. So clustered, regularly interspersed short palindromics repeats, CRISPR, originally evolved to be a defense mechanism in archaea and bacteria against virus invasion. Two distinct RNAs can activate the Cas protein and guide it to bind to the DNA sequence of the virus. And after binding, the Cas protein cleaves the viral genome and prevents replication. And various types of CRISPR systems have been identified and adopted for genome editing. So in general, the two RNAs are combined into a single guide RNA, or an sgRNA. Cas binds to the target DNA sequence using the sgRNA and undergoes a conformation change so that the RNA sequence binds directly to the target DNA sequence to allow cleavage. At this site of DNA cleavage, it can be repaired in such a manner that errors are introduced at that site, called insertion deletion mutations or indels. Or donor DNA can be introduced through the use of flanking homologous arms of DNA. This allows one to precisely change the DNA sequence at that location. There have subsequently been many advances in CRISPR gene editing. To name a couple, these include CRISPR-Hot, which has been described in organoid cultures. CRISPR-Hot allows precise integration of exogenous DNA sequences into a specific locus, but avoids extensive cloning and is reported to outperform homology-directed repair. The authors demonstrated this very nicely by using CRISPR-Hot to fluorescently tag various subcellular components and generate reporter organoid lines, which are these shown here. In addition, Cas9 has been modified by fusing an inactivated Cas9 nickase to cytidine deaminase to generate base editors. Both cytidine and adenine base editing have been described and can be used to generate precise changes in the genome in organoid cultures. There have also been multiple studies that have combined genome editing with organoid technology to model human disease. This includes studies that, for example, generated a frame shift mutation in the APC gene in GI organoids and showed that the organoids exhibited Wnt signaling independent growth. There have been a number of studies demonstrating the potential utility for CRISPR to correct monogenic disorders using organoid cultures, such as cystic fibrosis. And then multiple studies that have used CRISPR and organoid cultures to develop multi-hit oncogenesis models. So for example, targeting ABC, SMAD4, TP53, and KRAS colonic organoids, and evaluating the organoids for malignant phenotypes, such as growth factor independent growth, invasive behavior, development of aneuploidy, and importantly, drug responses. Some initial studies focused on generating organoids from genetically engineered mouse models, such as this study, which very nicely derived organoids from BRCA1 and BRCA2 deficient cancers in mice. Organoids retained features of the original tumor, such as DNA copy number profiles. They then orthotopically transplanted the organoids and found that they gave rise to mammary tumors that recapitulated the epithelial morphology and importantly preserved the drug response of the original tumor. And this is just one example of multiple studies that demonstrate the utility of organoids in establishing in vivo platforms for functional cancer genomics. Multi-hit oncogenesis was recently modeled in human breast organoids by performing targeted knockout of 4 tumor suppressor genes, P53, P10, RB1, and NF1. Notably, the authors showed that you can sort out the specific cell types that you're interested in transforming. Here they sorted these cell types from normal human breast tissues. And then they used CRISPR-Cas9 to perform sequential gene editing. They also used strategies such as treatment with NOT1-3A to select for organoids with P53 mutations. The authors then transplanted the human organoids into mouse mammary glands and showed that the more tumor suppressors that were altered, the more efficient tumor formation was. And the tumors modeled human breast cancer and also expressed ER and PR. They also took advantage of the different indels that could be present in different clonal populations to look at the clonal architecture of the tumors that were generated. There's also been interest in performing genetic screening in organoids. The goal here would be to knock out gene expression on a genome-wide scale and evaluate phenotypic changes using an sgRNA library that targets every gene in the genome. But you can also do this using a more specific gene set defined based on your parameters of interest. Multiple sgRNAs are used to target a single gene to reduce false positives, and this can be performed as an array or in a pooled fashion. There have been multiple challenges associated with adaptation of these techniques to organoid cultures, including that organoids are prone to false positives due to heterogeneous growth from organoid to organoid inherent in the properties of this type of growth from stem-like cells. Thus, high sgRNA abundance may be due to random outgrowth of any arbitrary organoid, and limited cell numbers can also be an issue, depending on the degree of any given line to exhibit clonal outgrowth. However, successful screens have been performed in organoids using limited gene sets of interest. In addition, techniques have involved transduction of single cells with growth of one organoid from each single cell, and then sgRNA representation is analyzed by sequencing each single organoid rather than the pool of all surviving organoids. Other approaches have paired each sgRNA to a random barcode so that you can identify single cell derived clones. And pre-screening approaches also have been performed using by performing the screen in cancer cell models first and then turning to cancer organoids. These advances are exciting, but only represent a small fraction of the different tools that can be combined to study breast cancer and breast cancer therapy using organoid cultures, and importantly, give you flexibility to tailor your approach to address your specific question of interest. So to summarize, organoid cultures are three-dimensional cultures that can be used to propagate diverse mammary epithelial normal and cancer cell types in culture. Organoids, as well as viable tissue, can be frozen to generate large biobanks for functional studies. Genome editing can be used to model tumor genesis, study repair of pathogenic mutations, or interrogate gene expression patterns at the cell or tissue level. And genetic screens in organoids can be challenging due to cell numbers needed as well as heterogeneous growth, but this can be overcome by using limited gene sets or by sequencing individual organoids, showing the potential of this technique for application to breast cancer. Here are my references, and I would also like to thank colleagues who contributed to some of the updated data in this presentation, and thank you very much for your attention.